From the RSNA, I'm Dr. Adeline Boettcher, and this is the Radiology Imaging Cancer Podcast. I currently serve as the scientific editor for this journal, and I'm very excited to bring you this new podcast focusing on preclinical studies related to cancer imaging. Radiology Imaging Cancer is developing a special collection on preclinical and translational cancer imaging manuscripts to highlight new advances that are being made in the field. Potential topics of interest include the use of animal and phantom models for evaluating the safety and efficacy of new imaging tracers, assessment of new imaging modalities and techniques, as well as many other exciting developments that are ongoing in cancer imaging research. Submissions for this special collection are now open, and the submission deadline is May 1st, 2021. Please see our website for details and reach out to editorial staff if you have any questions. To kick things off, this podcast will focus on 3 recent publications that leveraged animal models in the development and characterization of new imaging techniques. First, I will be speaking with Drs. Umar Mahmood and Benjamin Larimer about their work on the development of a PET tracer for granzyme B, which has really exciting applications in immunotherapy response assessment. Next, Dr. Nikita Consul, one of our trainee editorial board members, will be speaking with Dr. Eric Ahrens about his recent work of assessing oximetry response to CAR T-cell therapy in a glioma mouse model with fluorine-19 MRI, which was published in our journal in January. To wrap up our podcast, Dr. Wendy Tu, another trainee editorial board member, will be speaking with Dr. Dong-Hyun Kim about his group's recent publication in our journal, which focused on the development of Sorafenib-eluting microspheres and transcatheter arterial chemoembolization in a rat model of hepatocellular carcinoma. We're very excited to bring this lineup of research to you, and we hope that you enjoy. To start off our lineup of research topics, I have with me Dr. Umar Mahmood and Dr. Benjamin Larimer to discuss their work on the development and characterization of a PET tracer for granzyme B. To note conflicts of interest before we begin, Drs. Mahmood and Larimer are both co-founders, shareholders, and consultants to Cytosite Biopharma. Dr. Mahmood is currently the director for the Center of Precision Imaging within the Department of Radiology at the Massachusetts General Hospital. He is also a professor of radiology at Harvard Medical School. For many years, his research interests have revolved around early phases of translation of new imaging agents for individualized approaches to disease evaluation and to assess treatment efficacy. Dr. Mahmood currently has multiple NIH grants focused around the development of new imaging tracers. Dr. Larimer is currently an assistant professor at the University of Alabama, Birmingham. There, his lab works on the development of novel imaging probes for cancer characterization. Much of his work focuses on the development of new PET probes, such as those for targeting HER3, CD3, EGFR, as well as granzyme B. Dr. Larimer also has ongoing grant support from the NIH and NCI for the development of imaging techniques to assess cancer immunotherapy response. So to begin our discussion, can you both give us a brief summary of your project on developing and characterizing a PET tracer for granzyme B?
Sure. Well, thank you. Very excited to be on this podcast. And about 5 years ago, in the group, we were thinking about immuno-oncology and really realized it was an unmet need in terms of early assessment for response. And the current standard PET imaging tracers didn't meet that need. So we thought we would jump into the space and develop it preclinically. And it took about 150 mice for us to get the immune models working right. And our first target was CD3. And we started working on a CD3 imaging agent. And as part of that assessment, one of the targets we were looking at from a protein point of view is granzyme B, as we were doing a systematic characterization of our mouse models and response assessment. And what we realized is the dynamic range in granzyme B was much greater than that of CD3. And so we made a concerted effort to develop a granzyme B imaging agent. And we were very fortunate that we had already worked on 150 mice to get the models working. And so we saw that the granzyme B, we were able to predict which mice were going to respond and which mice weren't going to respond even before there were imaging changes. And so then we joined in with some leaders in immuno-oncology in the melanoma space at Mass General, Keith Flaherty and Genevieve Boland, and they provided us human samples. So that helped us correlate our findings preclinically with clinical response assessment. So that's sort of how we got into it. It was a little bit of luck and a little bit of targeted development.
Yeah, and just to add to that, I can remember back when people still met in offices. Umar and I and Eric Werenberg-Klee, who's a huge part of this discovery as well. We were sitting in the office looking at some Western blots, and we could see granzyme B was hugely upregulated, but it was an extracellular target, and canonically extracellular targets don't make good imaging agents. And we were having this robust debate, and I actually fell on the side that there was no way we were going to be able to image this. And Umar said, well, just give it a shot. And so we put together the probe and injected it into the first set of mice. And I've still got the text message I sent to Eric when I got the first images. I'm like, I can't believe it worked. And the rest from there was just downhill sledding. It's been a lot of fun, and we continue to enjoy seeing where we can push this thing.
Just to add to that, we were targeting on CD3, and I remember Ben and I were at one of the Society of Nuclear Medicine conferences. We were thinking about CD3, and Eric had said, kept talking about granzyme, and we were like, well, like as Ben said, it's extracellular, it's going to be hard, it may not work, but we tried it, and we've been very, very fortunate. It's worked out better than we ever expected.
Yeah, that's exciting to get things to work in a preclinical model. You have something that you can move forward with, which leads me to my next question is your groups at the University of Alabama and MGH are currently conducting a phase 1 clinical trial for this tracer, which was initiated in 2019. And the objective there is to use the PET tracer to predict immunotherapy responses in patients with melanoma and non-small cell lung carcinoma. So can you talk a little bit about what steps you had to take in translating this PET tracer from a preclinical to a clinical setting, and then what challenges you had to overcome?
Sure. I'll start with this one. The first real challenge is that there is no roadmap for when something should go from preclinical to clinical. And really, the way we went about looking at it was we talked to other people who had either done it before, or like Umar mentioned, Keith Flaherty, and the turning point for us was seeing in a meeting with Keith, and we were like, what else should we do? What else should we show to prove that this should go into people? And he said, this immunotherapy is such a new field, you just need to go into people. And so then the next question was, okay, how do we put together the package that the FDA needs to submit an IND? And that's the second big challenge, is that preclinical toxicology is a key and important step in being able to show something will be safe to go into people. But it's kind of there are funding mechanisms, but they're less well known, and there's a big gap for that. And so, fortunately, Umar has been doing a lot. He'd been doing a lot of preclinical to clinical translational work, and he had connections, and we were able to connect with a longtime biotech CEO named Jim Jensen, who was able to help guide us. And maybe I can let Umar tell more about that part of the story.
Yeah, so I'm a big believer in domain expertise, and that's sort of, if you look at sort of different areas that we all do, whether what Ben does, what I do, what Eric in the lab was doing, and the domain expertise extends to business and translation and fundraising and all of that. And the question is, when can you do it by yourself in a lab? And when we thought that this was very promising, and we saw this large unmet need. And so we thought, well, we could do it from an academic perspective and get that going, but that may take a long time and a lot of our bandwidth. And so when we decided to spin off a company and do it that way, and we went through and we're able to now do it at 3 sites. So the trial is open at 3 sites at MGH and Alabama. And actually we picked, because of the delays because of COVID, we actually opened a 3rd site in Taiwan. Because Taiwan has a lot fewer COVID cases than the United States. And so now the trial is open at both at Alabama and MGH, and we're fortunate we were able to recently image our first patient despite COVID, and we're about to image our second patient. So the first patient was recently done in Boston, second patient's about to be done in Birmingham. And so things are moving forward. And it was an interesting decision. There's a lot of stuff you can do in a lab preclinically. And when, I've held multiple INDs, but to translate it from a sort of not only first in human, but first in class tracer takes additional development work. And so we really thought the scope was beyond what an individual lab could do.
Yeah, it sounds very exciting to hear a little bit of a snapshot of, the beginning of a clinical trial. Usually you see the announcement and then like the final product, but kind of hearing the process that takes place behind the scenes is very cool. So talking a little bit about the applications of your PET tracer, I think granzyme B is a really exciting target because it's expressed by both CD8 T cells and NK cells. So in addition to potentially stratifying responders and non-responders to checkpoint blockade, you could also use this PET tracer for assessing cytotoxic activity of cellular cancer therapies as well. So can you give us your thoughts on using this PET tracer for monitoring adoptive cell therapies as well?
Yeah. We also identified that, and we have been working with collaborators out in Germany, a PI named Sebastian Kobold, who actually developed some adoptive T cell transfer models, and we've worked with him for several years now, showing that these T cells can specifically home to targeted tumors. We have a really cool model where on one shoulder is implanted a cancer that has a target, the other shoulder has a cancer that does not have the target. We can specifically delineate the T cells going to the target cancer and killing it. It's imaging, so you can do multiple time points and longitudinally watch as these T cells ramp up and they're killing. And we think that it holds a lot of promise for these up and coming adoptive cell transfer technologies.
Yeah, and one of the things we realized is because granzyme is a common downstream pathway for a lot of immune cell killing of tumors, that a lot of different approaches would work. So not only checkpoint inhibitors, and as we discussed, cell-based therapies, we also realized that tumor vaccines would potentially be imageable as well. And so we started doing some imaging a couple of years ago along those lines as well. So one of the nice things about granzyme B is that it is this common pathway. So it's sort of agnostic in terms of what cells are killing tumor cells, how those are killing it. So we thought that also increases the ability to assess some of these different therapies.
Yeah, those all sound like really exciting applications and leads to a lot of different projects that you could do in the future, which leads to my last question, which we kind of touched on is, what are some of the steps that your groups are taking to further develop this tracer or potentially other tracers for clinical use?
And so one of the things we realized is, as part of this development, that an academic center can develop certain tracers and certain approaches to some level, and then a company or a larger concerted effort can develop the next generation. So in addition to our gallium-based probe, we're also developing small molecules that are fluorinated. Our plan is to try this in a lot of different cancers. So right now, the first step is melanoma and non-small cell lung cancer. But you can imagine there are a lot of cancers. Every week we hear about a new cancer where checkpoint inhibitors are being approved. And so is to expand which cancers we're seeing to see how granzyme imaging might work, and then to try new probes to see how those might work as well. And so that's sort of been our approach, is how we're going to keep going and open up more sites. We right now are at 3 sites, but the plan is to make this available. I know that Ben and I truly believe in the granzyme. We've had thousands of discussions about this, and we really believe in this approach. And so our goal is to get it out there and see in other investigators' hands how they can take it for a road.
This has been my first clinical experience. I'm a PhD, and it just has given me such an appreciation for the clinical researchers, because when you do it preclinically, you can have a batch of 8 mice and you can have statistical analysis right away. But here, developing this clinically, you get your first patient, you look at it, and you don't know. It's an N of 1. You have no idea if that's good or is it bad. You have you can make some guesses, but it's truly a challenge, and one that I have a lot more appreciation for now going through it. That being said, because it's a slower process, we're always thinking ahead, and this first trial is focused on safety, but we're also looking at would baseline levels of granzyme B be predictive of who might be more responsive to immunotherapy? Umar kind of alluded to the fact that granzyme B sort of sums up everything that's going on with a tumor, and you could imagine if a tumor was a more inflamed tumor to begin with, it might have higher levels of baseline granzyme B. just something we've seen preclinically, but also very interested to look at clinically. Yeah, looking at beyond checkpoint inhibitors is another thing that's really interesting. For instance, in melanoma, we use a lot of TVEC here to try to potentially boost the immune response in melanomas. Radiation can obviously induce a lot of inflammation as well. And then chemotherapies, given at the right dose, there's questions now, what's the right timing if you're going to add an immunotherapy onto it? That it feels like the sky's the limit in terms of things we can do now that we have this into people. And it's just about finding more hours in the day to be able to get this going.
Yeah, I think we all feel that way when we're on an exciting track. So I want to say thank you both for sharing your experience. I think this was a nice taste of, how a tracer or really anything can, transfer from preclinical to clinical. And that's an interesting space for people to learn about. And we look forward to hearing about your future advancements in this area.
Well, thank you so much. Really appreciate the opportunity.
Yeah, really appreciate having the chance to talk to you today.
Next, we'll discuss another imaging application for monitoring responses to immunotherapy.
This is Nikita Consul from RSNA's Radiology Imaging Cancer, serving as a trainee deputy editor on the editorial board. I'm currently a radiology resident at Baylor College of Medicine in Houston, Texas, and will be doing a body abdominal imaging fellowship at MGH to commence July 2022. I do have a background in translational research during my years as a chemical engineering student at MIT when I worked extensively with murine models for nanoparticle drug delivery research in Dr. Sangeeta Bhatia's lab. And so I was interested in the article that we will be discussing today. In our January 2021 issue of Radiology Imaging Cancer, one of our featured articles is entitled Assessing Oximetry Response to Chimeric Antigen Receptor T-cell Therapy Against Glioma by Fluorine-19 MRI in a Murine Model, authored by Dr. Fanny Chapelin from University of Kentucky with principal investigator Dr. Eric Ahrens from University of California, San Diego. This study shows how F19 MRI can be used to measure tumor cell oxygen tension in response to chimeric antigen receptor T-cell therapy over time in murine models. This methodology can then hopefully provide insights into the mechanism of this type of engineered T-cell immunotherapy as it works against cancer, in this case, gliomas. I have with me today Dr. Eric Ahrens from the Department of Radiology at University of California, San Diego, and principal investigator for this study. Dr. Ahrens is the Director of Stem Cell Molecular Imaging and Professor of Radiology at UCSD since 2013. Previously, he served as professor at Carnegie Mellon University for 13 years, during which he briefly served as Chief Scientific Officer of CellSense. His background is in physics, and he earned a master's and PhD in physics at University of California, Los Angeles. Currently, he is the principal investigator for several NIH-funded grants, one of which is for compositions and methods for enhanced Fluorine-19 MRI cell tracking. He has 12 patents and over 150 peer-reviewed publications and abstracts, and we are honored to have him with us today. And Dr. Ahrens, thank you for joining us.
Yes, thank you. Thank you for the invitation to speak today.
My basic understanding of Fluorine-19 MRI is that this tracks a fluorinated contrast agent that can label cells without changing their architecture, and upon cell death, this label is released. I know that the use of MRI to track fluorinated organic contrast agents to track immunotherapeutic cells was first demonstrated by you, Dr. Ahrens, in 2005, detailed in your manuscript that was published in Nature Biotechnology. Do you mind briefing our listeners on how exactly Fluorine-19 cell labeling and tracking works and how MRI is used to monitor the cells?
Yeah, of course. The basic idea that we conceived around 2003, 2004 was that we wanted to move away from contrast agents and using proton scans because there's some ambiguity often as to where if cells are present or not in tissue. There's a possibility for false positives, false negatives, particularly due to the intrinsic contrast that you have present in in organisms. So we want to go with a pure tracer material where instead of looking at the indirect effect of the contrast agent on surrounding water, we look and image directly the material that we put in, so-called tracer material. Inspection of the periodic table suggests that fluorine 19 would be a suitable candidate for a tracer atom due to its high gyro-magnetic ratio and hence sensitivity in MRI, its spin 1/2, and its known uses in biomedicine in previous applications. So we developed formulations of perfluorocarbon oils, which are highly fluorinated molecules with a carbon-fluorine bond, a lot of chemically equivalent fluorine atoms so that you get a single NMR resonance. We formulate those as an emulsion, i.e. a colloidal suspension with droplets about 150 nanometers in diameter. This colloidal suspension is stabilized with a surfactant to ensure self-life. And you can engineer the surfactant so that it can be taken up by cells in culture. So you would add the emulsion to cultured cells, for example, part of a cell therapy or immunotherapy, for example, T cells or stem cells. And then with co-incubation, you get an uptake of the agent inside of the cells. And this can take 3 hours or overnight. You do a wash step, and then you can inject the cells into the subject and then image the cells at multiple time points using fluorine-19 MRI. You also take a proton scan, conventional scan in the same session that's registered. And so you get the hotspots from the tracer in the anatomical context. That's the basic idea. We like the fluorine because there's no background signal. There's essentially no false positives. What you see in the hotspots is what you put in the body. Thus, the interpretation of the images is straightforward. The type of imaging that we do, MRI, is spin density weighted imaging, the simplest form of MRI, I would say, where the amount of signal that you get out is proportional to the number of fluorine atoms that are present, linearly proportional. And so you can do quantification in a way that's straightforward and doesn't require modeling. And so quantitative readouts is, with unambiguous detection, are two desirable features that we've achieved with this technology.
Thank you, Dr. Ahrens. And now coming to the paper that we are featuring in our journal, we would like to hear from you in your own words, a summary of the exciting findings from your group's investigation that led to this paper based on the methodology that you just explained.
Sure. So building on the methodologies that I described, we asked the question is since we have a perfluorocarbon droplets inside of the cell, can we use that as an intracellular sensor? And our understanding was that the perfluorocarbons had a long history in use in biomedicine, particularly in the context of artificial blood substitutes, which I think there was a lot of investment made. During the HIV/AIDS scare in the late 80s, there was a lot of effort trying to develop, there was concerns about the safety of the blood supply. And so there was efforts to develop artificial blood so that you could bypass these safety concerns. And so perfluorocarbon emulsions was considered a candidate. So oxygen readily dissolves into the fluorous phase, into the droplets. And you can dissolve quite a bit of oxygen in there. And so you can serve as an oxygen carrier. What we also, other people have realized is that the oxygen that's dissolved in the fluorocarbon affects the NMR properties of the fluorine-19. So oxygen is weakly paramagnetic due to the singlet oxygen, presence of singlet oxygen in minor amounts that is dissolved. This paramagnetic species then has a through-space interaction with the fluorine nucleus and causes a shortening of the T1 relaxation time, fluorine-19 T1 relaxation time. And the amount of shortening is linearly proportional to the partial pressure of oxygen that's dissolved in the fluorous phase. And so if you can measure T1 in situ, you can measure then with a lookup table the PO2 in absolute numbers in millimeters of mercury, for example. And so you have a true intracellular oxygen sensor using this technology. So you can then label your cells ex vivo, put them into the subjects, and then you can measure multiple times T1 in vivo, and then monitor the time-lapse PO2, real-time PO2 measurement. And the perfluorocarbon persists inside of cells as long as it remains viable. And so for the lifetime of the cell, and if the daughter cell, even daughter cells in dividing phenotypes will also harbor the probe. The T1 measurement is independent of the concentration that's present within the cell. And that's also a very helpful feature. So even if there's cell division, you'll still get a T1 measurement that's reliable. So the idea is that in the paper, what we did then, we wanted to apply this to an immunotherapy model that's contemporary. So we looked at a xenograft model of a U87 human glioma cell line that constitutively expresses EGFR variant 3. And then we had a human T cells that we obtained from the blood bank. We transduced them with a lentivirus to express the EGFR variant 3 CAR receptor on the surface of the T cells. And that's basically our model. And in the paper, we independently label both the glioma cells ex vivo before implantation into the flank of a mouse, or the T cells we labeled before implantation into the same U87 model. We looked at both sides. And then we looked at then what happens when you have a cell-cell interaction between the effector cell, the CD8 positive CAR T cell, and the glioma cell. How does that change PO2, looking at both from the perspective of both the glioma cell and the T cell? And the key finding we obtained was that when looking at the PO2 inside the glioma cells, you get a delayed but peak in the PO2. So PO2 goes up in cells that are becoming apoptotic. And this has been observed by our group before. What's interesting about this model is that the increase in PO2 inside the glioma cell is delayed by about 3 days. It peaks, and then it goes down again by about 7 days. So it's not a sustained effect. So for example, if you were to inject chemotherapy that would kill the glioma, you'd have an increase in PO2, but it would be sustained for many, many days. So, but with the immunotherapy, it comes and it goes. And so this suggests mechanisms of, or the modes of action of the effector cells. And perhaps, for example, that there's thoughts that you could have a T cell exhaustion, for example, that would limit the killing ability of these cells. Or there's other hypotheses. But this is a way that you can look at in real time, the efficacy of the tumor killing, and then hopefully design co-therapies, for example, things that you could add along with the T cells that could allow you to have a more sustained PO2 response, i.e. killing response with the same dose. Or it could also suggest that you need to put in multiple doses of cells in order to get a sustained effect. So this is an interesting area that had never been measured before. We also found that when we label the T cells and then we put those into the tumor, that we didn't get really a pronounced peak in PO2 at all. It remained pretty flat. And so that suggests that either the T cells can regulate oxygen much better than the glioma cells are able to when they're being attacked, or it could mean that the T cells are just heterogeneous. There's also mixed in CD4 cells and things like that, and that can sort of dilute out the signal. So anyway, this opens up a lot of possibilities for a real detailed metabolic look in real time of what's happening with T cell therapies in real time in individuals. And anyway, it's been stimulating a lot of new efforts in our lab.
Thank you for that explanation, Dr. Ahrens. Indeed, this is sounds like an exciting type of murine model for studying immunotherapy tumor cell treatment mechanisms in real time, as you said. What are the follow-up questions or research ideas, future investigations that your lab has planned as the next steps?
Yeah, I mean, this suggests a lot of interesting with these tools that we develop. We're kind of tool makers. And so we've developed a novel way to see something new. And that's always a lot of fun when you come up with a new tool and you can see new biology. And so the obvious things to try would be, can we get a more sustained PO2 response from the tumor killing? Make it look more like what happens when you add a systemic chemotherapy, for example, where you get a sustained killing effect. So this can involve co-therapies, for example. For example, antibody therapies, if you add that in the mix, will that give you a more sustained PO2 response? Or if you redose the animals. Or, for example, you could precondition the tumor, for example, with radiation, and will that give you a more sustained effect? So there's all these different ways, and what's nice is we can read out the response sort of in real time and study the same animal over time. And sort of investigation of co-therapies would be an interesting direction.
Yeah, this sounds all very interesting. I mean, it could completely change the way we think about cancer treatment in the future and how we plan cancer treatments for individual patients. It was really great to hear about these future directions from you in your own words, Dr. Ahrens. I would like to now focus specifically on the murine models that your group used in these investigations as a conclusion to this interview. What are some of the challenges that you faced, that your lab faced using murine models, or any other aspects of the translational methodology that were used in this research?
Yeah, I mean, we're working on treating solid tumors with immunotherapy or T cells, and this has run up to limited success, I believe, both in the animal studies and also in the clinic. These are xenograft models, so the immune system behaves differently than humans. The other thing that we run up against, the main obstacle we run up against, is that the amount of cells that actually traffic to the tumor are very low, right? We put in maybe 10 million cells. In a prior study, we looked at actually how many cells actually make it to the glioma after about 7 days. And it's only something of order 50,000 cells total with 10 million put in. So it's a very inefficient homing. So that's one of the main limitations that we have with animal models. You don't put a lot of cells in comparison to the body that you would in humans. The tumor size is about the same or same order of magnitude, and the homing efficiency is low. And so it doesn't recapitulate the human scenario very well. And so that's, the model that we use is not the most current state of the art, I would say. It's maybe a few years old. It's probably a typical problem that people run against. When you're talking about immunology and comparing it to humans, there's always these limitations in these type of models. And that's why they call them models. They're not the real thing. And so the limited tumor homing is the main issue.
That was very helpful to me and our listeners, Dr. Ahrens. Thanks again for joining us on this podcast episode.
Thank you. And I'd like to just thank Fanny Chapelin, this is the bulk of her PhD thesis in my lab, and she did a tremendous amount of work to produce this. And so I'd just like to give my gratitude for that. Thank you.
Yes. Congratulations once again to you, Dr. Chapelin, and the rest of your group for these developments. We look forward to seeing what direction your group's work takes in the future. For our listeners, thanks for tuning in for today's episode. We look forward to having you join next time.
For our last session, we'll learn about an interventional radiology application that was tested in a mouse preclinical model.
All right, hello everyone. My name is Wendy Tu. I am a body interventional fellow at the Brigham and Women's Hospital, Harvard University in Boston, and I'm also a member of the trainee editorial board on radiology, imaging cancer. As part of this episode's focus on translational imaging techniques and animal models, I have the pleasure of interviewing Dr. Dong-Hyun Kim on his recent publication in Radiology, Imaging Cancer entitled Development and Validation of Sorafenib-eluting Microspheres to Enhance Therapeutic Efficacy of Transcatheter Arterial Chemoembolization in a rat model of hepatocellular carcinoma. Dr. Kim is the corresponding author and an associate professor of radiology and biomedical engineering at Northwestern University. He is also the director of biomaterials for imaging-guided Medicine Lab at Northwestern. He has an impressive resume with many research interests, including image-guided medicine and new cancer therapies, which can overcome the limitation of current conventional therapies. He has also been studying multifunctional carriers to treat cancers for 15 years and has published over 60 papers on international refereed journals. Thank you for being here with us and welcome.
Thank you. Thank you for having me.
As we know, transcatheter arterial chemoembolization, or TACE, is a widely employed treatment for non-operative hepatocellular carcinoma, and it's been backed by extensive literature demonstrating increased overall survival compared to conservative approaches. I read your study with great interest, especially in the use of an animal model and imaging with iron oxide particles. My first question for you is, what inspired you to undertake this project?
Okay. Yeah. First of all, as you know, that the hepatocellular carcinoma is one of the very serious cancers. So that motivated us to study about the treatment of method. So you know that the HCC is really very common, and the mortality is pretty high. So that is like the first leading cause of cancer death in the U.S. And also the more importantly, like the developed countries also have a significant increase of the HCC incident rate over the last 15 years. So it is pretty significant cancer and need attention for the treatment of the HCC. And then now that the current therapeutic options for the HCC is that the resection or transplantation, that would be the potential curative that the treatment method, but it's only very small portion of patients are eligible. So except for that is you know that the Sorafenib multi-kinase inhibitor, that is the first line of the HCC treatment. Yeah, that has been used, but survival benefit is not really great. So we are thinking about what could be the limitation, and then the important limitation is coming from that the systemic application of the Sorafenib. So those systemic dose to induce the severe toxicity, and then that can finally result in the low overall therapeutic response. So that we have been thinking about the local delivery of Sorafenib that can be more effective with minimizes systemic exposure. So we developed some of idea for the combining the C-TACE, conventional TACE, and then Sorafenib-eluting microspheres.
That's fantastic. And I also had a question for you regarding working with an animal model. How do you find this differs from doing clinical trials in humans? And how can we translate these results from animals to humans?
Okay, so this animal model is a HCC, that the rat animal model has been used a very long time. So I think to evaluate those interventional oncology procedure, the rat animal model has been used. So, and also those are currently available that the commercial drug eluting bead has been tested in those kind of rat animal models. So that we are thinking that animal model can be good for the translation. With the confirmation of those effectiveness of those interventional oncology therapeutic and then procedure, maybe we are thinking that can be really translatable.
For sure. And you mentioned drug-eluting beads, which are a very cool piece of technology. What sort of main advantages does drug-eluting beads offer over the traditional delivery systems?
The drug-eluting beads actually that has been used for that TACE, drug-eluting bead TACE. So drug-eluting bead can encapsulate various kind of anti-cancer chemo agent, and also depending on the material of the microsphere and the design of the microsphere, the pharmacokinetics of those loaded chemo drug can be controlled as we desired. So especially those advanced advancement of the recent chemistry and nanotechnology, there are the many variables for loading and the delivery of the various cancer chemo agent. So also the possibility other functional agent can be added in those kind of drug-eluting microbead, and also those such like a MR imaging agent as we use in this paper, and also like a CT contrast agent also can be used for the drug-eluting bead. So like in our the paper, we used the iron oxide nanoparticle, and also that in case of the embolization, that the application, depending on the drug-eluting bead size, so we can control the location of embolization and also location of drug source as it can be controlled. So that means we can control the embolic effect with the drug-eluting bead. So I think that there is a really big potential to be developed for various kind of drug delivery application, especially when we need those kind of local delivery of the anti-cancer agent. So actually currently there are many kinds of the commercial drug-eluting bead, but the further it is expected more advanced drug-eluting bead will be developed for various interventional oncology procedures.
That's a great point. And in terms of using sorafenib, how does sorafenib compare to traditional chemotherapeutics agents that were previously used, like doxorubicin?
So doxorubicin is a cytotoxic drug, so which can kill cancer cells directly with like a DNA intercalation, and also that can induce the cancer cell apoptosis. But the sorafenib is also a little bit different. So sorafenib also can have the cytotoxic effect, but in addition to that, that has multi-kinase inhibitor function. So that can work for various other pathways, is focusing on that angiogenesis and then tumorigenesis. So the people knows that the sorafenib is primarily the working for the VEGF inhibition and anti-proliferation of the tumor. So I think that the main target of each drug, doxorubicin and sorafenib, is different. So and also that the further, if we're thinking of the two different properties, so when those two agents can be combined, we could expect the more synergistic effect to killing the cancer cell. So the recent clinical trials have been ongoing for combining those TACE and the sorafenib. So those two combinations, sorafenib and the dox-TACE, can have more synergistic effect to kill cancer cells in vivo.
You mentioned sort of previous literature looking at sorafenib and TACE. How does your research differ to comparing to the previous randomized controlled trials of the combination of sorafenib and TACE in humans?
Yeah, actually, those trials there is the same combination, but in the trial, the sorafenib has been applied with systemically. The sorafenib has been taken by the oral, so that means the sorafenib is can applied systemically. So one of the main the limitation with sorafenib is the side effect. So even if we can expect the synergistic effect of doxorubicin and the sorafenib, but the limitation of the side effect of sorafenib is still there. So yeah, that's why we propose the sorafenib-eluting the microbead that can deliver sorafenib in locally. So we are expecting those differences can make a better outcome compared to the current clinical trial.
That's fantastic. Do you anticipate this treatment to be applicable to all patients, or will we have to select our patients carefully when applying this treatment?
So this procedure actually based on the standard TACE. So I think as TACE is performing in clinic with the specific patients. So those criteria can be applicable for this procedure as the same. So just one minor concern would be there is no clinical data about the local delivery of the sorafenib in the tumor area or IA delivery of the sorafenib. Those data is not tested actually. So in our preclinical animal test and the study, there was no problem with the IA infusion of the sorafenib in the safety and the therapeutic efficacy, but it should be confirmed or it should be tested.
That's a great future direction that you mentioned there. What do you think was the most challenging aspect of this project?
One of the challenging things on this project was the animal study. So actually, this is a preclinical study using the hepatocellular carcinoma rat model. So, and then we need to do the hepatic intra-arterial infusion procedure for infusing the lipiodol mixed doxorubicin and the sorafenib-eluting microsphere. But we used a very small small animal rat. So that means that the hepatic artery is really small. So those are like the intra-arterial infusion technique has been really challenging. So yeah, that was the main challenge. But in our lab is that we have been done those procedure so long time, and then we have a pretty well trained investigators is here, so we could be successful. But those procedure itself can be challenging in those small animal model.
Wow, yeah, that does sound very challenging. A very small hepatic artery, yeah, for sure. Wow, so thank you so much for taking the time out of your busy schedule to join us today and talk about your fascinating paper. It's been a real pleasure to speak with you today. Thank you again, Dr. Kim.
Thank you very much. Thank you for having me.
Of course.
Thank you for joining us today. We hope you enjoyed this overview on recent developments in preclinical imaging. We're always happy to hear any feedback and potential topics of interest for the future. Please subscribe on wherever you are listening. Be sure to check out our upcoming preclinical works as we add to our collection later this year. I'm Adeline Boettcher, and this is Radiology Imaging Cancer. See you next time.